Hello, and welcome to G Fun Facts Online. Today, we are diving deep into a microscopic revolution that is fundamentally changing how we understand the building blocks of life. We are talking about acoustic nanoscopy and the incredible science of utilizing guided waves for single-cell diagnostics. Now, when most of us think about cells, whether it is a red blood cell, a skin cell, or a neuron, we tend to visualize them as we saw them in high school biology textbooks. We picture static, frozen blobs. We imagine them as structural building blocks, like bricks in a wall. But this image is fundamentally incomplete. In reality, the cellular universe is a noisy, vibrating, dynamic place. Cells are not static. They are constantly stretching, stiffening, relaxing, and oscillating. They vibrate in response to their environment. They hum with metabolic activity. For decades, medicine has relied on seeing these cells. We use optical microscopy, we stain them with dyes, we tag them with fluorescent markers, and we look at their shape and size. While these visual methods have revolutionized biology, they miss a critical property of life: mechanics. A cancer cell is not just a misshapen blob, it is mechanically different. It is often softer than a healthy cell. A red blood cell infected with malaria loses its elasticity long before it shows a visible defect. To truly understand health and disease at the most fundamental level, we need to stop just looking at cells and start listening to them. This is the frontier of acoustic nanoscopy. It is a field that uses high-frequency sound waves, specifically guided acoustic waves, to probe the mechanical soul of a single cell. So let's get into the physics of this. How do you listen to a cell that is only a few micrometers wide? We aren't talking about the kind of ultrasound you might see in a pregnancy scan. Traditional ultrasound provides grainy, black and white images of organs. Acoustic nanoscopy operates at a completely different scale, using frequencies in the gigahertz to terahertz range. At these incredibly high frequencies, sound waves have wavelengths that are comparable to the internal structures of the cell itself: the nucleus, the mitochondria, and the cytoskeleton. By harnessing what we call guided waves, acoustic energy confined within thin layers, or traveling along specific pathways, scientists can now measure viscosity, elasticity, density, and compressibility with nanometer precision. And the best part? They can do this without ever touching or labeling the cell. To really grasp this, we have to abandon our everyday understanding of acoustics. In our macro world, sound travels as a bulk wave through the air, but when you shrink the scale to nanometers, sound behaves differently. This brings us to the concept of guided waves. In traditional bulk ultrasound, energy spreads out in all directions, losing intensity. A guided wave, however, is confined within a specific boundary, like a waveguide. Think of a fiber optic cable carrying light over long distances without losing the signal. Now imagine that same concept for sound. In acoustic nanoscopy, the waveguide might be the surface of a microfluidic chip, a thin polymer layer, or even the cell membrane itself. There are three main types of waves we need to talk about here: Rayleigh waves, Lamb waves, and Love waves. Rayleigh waves are surface acoustic waves that travel along the surface of a solid. Their energy is confined to a shallow depth. In cell diagnostics, these are often used as invisible tweezers to manipulate and sort cells. Then you have Lamb waves. These propagate in thin plates, like the wall of a microfluidic channel. They are incredibly sensitive to changes in mass and stiffness. When a single cell touches a sensor surface, it changes how the Lamb wave travels, allowing scientists to measure how sticky or stiff the cell is. Finally, and perhaps most critical for diagnostics, we have Love waves. These are sheer horizontal waves trapped in a thin guiding layer. Because of their sheer motion, they don't lose much energy into the fluid surrounding the cell, which makes them incredibly sensitive biosensors. A Love wave sensor can detect minute changes in the viscosity of a cell's cytoplasm as it reacts to a drug. But there is an even more elegant method of listening to cells, one that uses light to detect sound. It is called Brillouin spectroscopy. This technique relies on something called spontaneous Brillouin scattering. It turns out that photons from a laser beam can interact with the natural thermal vibrations inside a cell. These vibrations are essentially fleeting sound waves created by the heat and movement of molecules. When a photon hits one of these moving sound waves, it gains or loses a tiny amount of energy. This results in a color shift, known as the Brillouin shift. This frequency shift is directly proportional to the stiffness of the material. So by scanning a laser across a cell, scientists can build a 3D map of the cell's stiffness. They can see a rigid nucleus or a soft cytoplasm without ever touching the cell physically. It is like using a laser as a remote microphone. For the highest possible resolution, scientists turn to something called picosecond ultrasonics. This sounds like something out of Star Trek. It uses femtosecond laser pulses, that is 1/4,000,000th of a second, to instantly heat a tiny spot on a cell or a transducer. This rapid heating causes a sudden thermal expansion, which launches a sound wave with frequencies up to several terahertz. As this ultra-high frequency wave travels through the cell, it bounces off organelles and interfaces. By timing these echoes, researchers can map the cell's internal structure with a resolution down to tens of nanometers. It is essentially sonar, but on the scale of a single cell. Now, why does all this matter? Why do we care if a cell is stiff or soft? This brings us to the biology of mechanotransduction. We used to think biology was all about chemistry, proteins and DNA, but we now know that how cells sense and respond to mechanical forces is central to life. Let's look at cancer. Cancer is fundamentally a mechanical disease. The cytoskeleton is the scaffolding of the cell, made of actin filaments and microtubules. Healthy cells have a rigid structure. But metastatic cancer cells often remodel this scaffolding. They depolymerize their actin network, making themselves softer. Why? Because this softness allows them to squeeze through tight gaps in blood vessel walls and invade other tissues. Acoustic nanoscopy can detect this softening long before a pathologist could see a change in shape under a microscope. A guided wave scan could theoretically grade a tumor biopsy by the stiffness of its individual cells, predicting exactly how aggressive the cancer is. It is not just about stiffness, though. It is also about viscosity. The inside of a cell is a crowded, thick soup of proteins and organelles. Changes in this internal viscosity often happen before structural changes. For instance, when a cell is dying, a process called apoptosis, the cytoplasm condenses and becomes more viscous. Love wave sensors are particularly good at measuring this. As the guided wave travels under the cell, the viscous drag of the cytoplasm dampens the wave. It gives a real-time readout of the cell's internal thickness. This technology also allows us to measure density and compressibility. Acoustic nanoscopy is one of the few techniques that can measure the mass density of a single living cell. This is significant because the density of a cell changes during its life cycle. Just before a cell divides, it swells up and its density drops. Monitoring these fluctuations can reveal the metabolic state of the cell and how it reacts to drugs that are supposed to stop cell division. Let's move on to the practical applications. How does this help patients? The transition of acoustic nanoscopy from physics labs to medical diagnostics is driven by its ability to act as a label-free biomarker. We don't need expensive antibodies or toxic dyes. The cell's physical state is the label. In cancer diagnostics, finding circulating tumor cells, or CTCs, in the bloodstream is like finding a needle in a haystack. Acoustic flow cytometry can separate these rare cancer cells from billions of blood cells based purely on their unique size and compressibility. It is a non-invasive liquid biopsy. In hematology, this tech is a game-changer for diseases like malaria and sickle cell. Red blood cells need to be incredibly flexible to squeeze through your body's tiniest capillaries. The malaria parasite invades these cells and stiffens them so they don't get filtered out by the spleen. Acoustic nanoscopy can detect this stiffening at the single cell level way before the parasite is visible. Similarly, for sickle cell disease, acoustic sensors can monitor how effective a treatment is by checking if the red blood cells are staying flexible. This also applies to stem cells. Stem cells hold the promise of regenerating damaged tissue, but we have to make sure they turn into the right kind of tissue. As a stem cell turns into a bone cell, it gets stiff. If it turns into fat, it stays soft. Guided acoustic waves can monitor a stem cell culture non-invasively, ensuring the differentiation process is on track without having to destroy the cells to test them. Then there is drug screening. Developing new drugs is expensive. Many chemotherapy drugs work by freezing the cell's cytoskeleton to prevent it from dividing. Acoustic nanoscopy can screen thousands of compounds rapidly to see which ones effectively alter cell stiffness. It is a high-speed functional test that could save billions in drug development costs. But acoustic waves don't just measure, they can also manipulate. This brings us to the field of acoustofluidics. By setting up a standing wave where two waves collide and cancel each other out in specific spots, scientists can create pressure nodes. A cell can be trapped in these nodes, levitated, and rotated in 3D space. This acts like acoustic tweezers. It allows for full 3D tomography, imaging the cell from every angle without any physical contact that might damage it. This is also used for sorting. A microfluidic chip can take a blood sample and acoustically push white blood cells into one channel and red blood cells into another, purely based on how they react to the sound field. This is critical for preparing samples for genetic analysis, where purity is key. Of course, all of this generates a massive amount of complex data. A Brillouin spectrum, or a picosecond echo train, contains subtle patterns that are invisible to the human eye. This is where artificial intelligence enters the picture. Algorithms like principal component analysis are used to deconstruct the complex signals, effectively separating the acoustic signature of the nucleus from the cytoplasm. Deep learning models, specifically physics-informed neural networks, are being used to solve the inverse problem, taking the raw scattered wave data and reconstructing a high-resolution map of the cell's mechanical properties. We are even seeing the rise of acoustic morpholomics. Just as genomics studies genes, morpholomics studies cell shape and structure. AI models are being trained to recognize the acoustic fingerprints of disease. In the future, an AI could analyze an acoustic scan and say, with 99% confidence, that specific cells are early-stage leukemia, based on their stiffness-to-density ratio. Companies like DeepCell are already pioneering AI-driven morphological sorting, and acoustic equivalents are close behind. This is not just academic theory, it is a burgeoning industry. Millipore Sigma acquired Flow Design Sonics to use acoustic cell processing for CAR T cell therapy manufacturing. Their systems use acoustic waves to wash and concentrate cells, which is a critical step in creating personalized cancer treatments. Thermo Fisher Scientific has the Attune Flow Cytometer, which uses acoustic focusing to align cells in a laser beam, allowing them to run samples 10 times faster than traditional machines without clogging. Then you have startups like Anitis Technologies developing acoustic beads to sort fragile cells gently, and hardware companies like Light Machinery building the ultra-precise spectrometers that make Brillouin microscopy possible. However, there are challenges. High-frequency sound is heavily absorbed by water. This limits how deep we can see. Currently, we can only image single cells, or very thin tissue slices, not deep inside the body. There is also the issue of heat. The energy required to generate high-resolution signals can heat up the cell, so balancing signal strength with cell survival is a constant engineering challenge. And speed is an issue. Scanning a single cell pixel by pixel can take minutes. For clinical use, that needs to drop to milliseconds. Looking to the future, the ultimate diagnostic machine will likely be a hybrid. Imagine a device that uses acoustic nanoscopy to measure stiffness, Raman spectroscopy to identify chemical composition, and AI to integrate it all. A single drop of blood enters, and minutes later, a complete mechanical and chemical profile of every cell is generated. Acoustic nanoscopy represents a fundamental shift in medicine. It moves us from a visual understanding of biology to a mechanical one. By listening to the guided waves whispering through our cells, we are uncovering a new layer of biological truth, one where the stiffness of a membrane or the viscosity of a nucleus tells a story of health or disease that no microscope could ever see. As physics, engineering, and AI converge, this silent technology is poised to make a resounding impact on the future of healthcare. Thank you for listening to G Fun Facts Online, and stay tuned for more deep dives into the science shaping our world.